Hi, my name is Ayushi Ahuja, and today I'm going to talk to you about GERD, or gastroesophageal reflux disease. This presentation will focus on the underlying causes behind GERD, which will then be critical in exploring promising research initiatives aiming to improve GERD treatment and quality of life. So my presentation is called The Word on GERD, and we will be understanding the causes behind gastroesophageal reflux disease in order to spur further exploration of precise treatments. Before we get into my project, here's a little bit about myself. My mentor for this project is Maria Broward. I'm a high school senior from Dallas, Texas, and I have a long-standing passion for biology research. I chose to do this project on GERD since it is a disease that runs in my family. This family connection motivated me to research more about the causes and new developments in the treatment landscape for GERD. I'm part of the speech and debate and HOSA teams at my school, and I enjoy playing the piano, traveling, spending time with family, and reading. A fun fact about me is that I am CPR certified. Now let's go over the research question we will be answering in this presentation. After an extensive literature review, we will understand how genetic and environmental influences lead to a diagnosis of GERD and the presentation of typical symptoms that are associated with the disease. Before we delve into the science behind the causes of GERD, let's define and understand what GERD really is. GERD is an acronym that stands for gastroesophageal reflux disease. It is one of the most common digestive disorders, with more than 3 million Americans diagnosed with GERD every year. As you can see, GERD is an important health concern and therefore is a priority for research and treatment. The Montreal Consensus of 2006 outlines a globally acceptable and standard definition that defines GERD as a, quote, condition that develops when the reflux of stomach contents into the esophagus causes troublesome symptoms or complications. Typical symptoms observed in common presentations of GERD include heartburn, acid regurgitation, and extraesophageal symptoms such as chest pain, nausea, and chronic cough or asthma. Risk factors associated with GERD are far and varied. Some are linked to lifestyle choices, while other risk factors are innate and uncontrollable. Firstly, age. An increased risk of GERD has been found in patients as age advances. With regard to gender, women are more likely than men to be diagnosed with GERD. Obesity also has a strong association with GERD symptoms, as excess weight puts more pressure on your stomach and digestive organs. Unhealthy dietary preferences and bad nutritional habits have been found to lead to an increase in the development of GERD as well. Physical activity is a protective mechanism against GERD, so low physical activity would be a risk factor. Increased physical activity keeps the body at a healthy weight and promotes proper functioning. Smoking and alcohol use have been both found to increase acid production, thereby increasing GERD symptoms. Certain medications that affect esophageal motility have also been associated with GERD development. And finally, childhood GERD has been found to persist through adolescence and adulthood in otherwise normal children. Let's take a look at the prevalence of GERD, which will help us better understand any skewed or uneven distributions. Now, GERD is an increasingly common gastrointestinal disease. The recorded uptick in patients diagnosed is a cause for concern amongst patients and researchers around the world. GERD is increasing in prevalence worldwide and is estimated to affect up to 20% of the global population. One point to note is that the Western world appears to have higher rates of GERD diagnosis, with a reported prevalence of 30% in adults within these Western populations compared to a prevalence of below 10% in East Asia. The map on the right side of this slide does a good job displaying these global prevalences by country, clearly showing us higher rates of GERD diagnoses in North and South America and the Middle East, and lower rates in Asia overall. These locational discrepancies can be due to genetic factors or lifestyle and health habits, which are influences that we will talk about in the coming minutes of this presentation. The final goal of this Polygence project is to write a literature review paper on the risk factors associated with GERD, with a heavy focus on the genetic risk factors. Existing literature will be reviewed in order to understand the genetic and environmental influences behind common presentations of GERD. Current research on 4 genes that are associated with GERD will be summarized so that we can understand any genetic correlations. For the purposes of this presentation, we will only be covering 2 of these genes, but my research on the remaining genes can be found in my upcoming paper. Additional discussion in my paper will be about environmental factors such as age and diet. Existing treatments will also be summarized with emphasis on those that target the genetic causes, and future research directions will be suggested to further advance the knowledge of GERD risk and improve treatments. Now, let's dive into the genetic factors behind GERD diagnosis. In my research paper, I performed a literature review on 4 genes linked to GERD: GNB3, ADRB2, BARX1, and ADAMTS17. But in this presentation, let's just take a look at the last 2 GERD risk loci. First, let's take a closer look at the BARX1 gene and its link to GERD. The BARX1 gene codes for a member of the BAR subclass of homeobox transcription factors. Animal modeling studies have indicated that the transcription factor that is encoded by the BARX1 gene may play a role in teeth development and the growth of complex structures in the skull and face. Relevant to GERD, the expressed protein has also been associated with the differentiation of epithelial cells in the stomach. The model to the right shows the function of the BARX1 gene in stomach epithelium development. Now, a 2019 cohort study conducted by Ahuja et al. Found a considerable association between the expression of BARX1 and GERD. After the researchers conducted lab techniques such as DNA extraction and allele-specific PCR, analysis of their results for the selected polymorphism, which is a type of DNA variation, showed that the frequencies of the AG and GG genotypes and also that of the minor allele G were significantly increased in patients that had GERD compared to the healthy controls. This finding helped researchers conclude that the study participants exhibited an association between BARX1 and GERD development. BARX1's role in GERD development may be related to the function of its expressed transcription factor in esophageal differentiation in the stomach mesenchyme tissue. The dysregulation or impairment of BARX1 could lead to a reduction in anatomical anti-reflux mechanisms, hence explaining the link between the BARX1 gene and GERD. Now let's move on to the second gene we will talk about today, ADAMTS17. The ADAMTS17 gene is a protein coding gene that encodes a member of the ADAMTS protein family. ADAMTS family proteins are cell surface proteins that contain sheddase functionalities. This means that ADAMTS proteins can regulate the shedding of important molecules in cells, such as membrane-bound proteins or growth factors. This function can promote a variety of health issues such as breast cancer cell growth and Weill-Marchesani syndrome, which is a rare genetic disorder. Now, ADAMTS17 belongs to a subgroup of orphan enzymes, meaning that its function has yet to be clearly defined, but we know that its unique structure with both adhesion and protease domains, as well as its involvement in many biological processes, is definitely of importance in GERD-causing mechanisms. Here you can see a structural model of the ADAMTS17 loci that shows the separate domains of the gene. The 2019 Ahuja et al. Study discussed previously also investigated a correlation between ADAMTS17 and GERD. A genotypic analysis was conducted on study participants that included some who were suffering from GERD and the others who were simply controls. The results of this experiment showed that patients with the homozygous GG genotype of the selected polymorphism had a 3.42-fold increased odds of developing GERD compared to those who carried the TT genotype or those with the presence of only one G allele. Understanding the functions of the ADAMTS17 gene, as well as the proteins it expresses, are key to uncovering the reasoning behind this correlation. One theory is that ADAMTS17's expressed protein has the ability to confer different regulatory influences on microfibrils in the cell environment. However, further research is still necessary to confirm the role of ADAMTS17 in GERD development. Moving on from genetic factors, let's now talk about environmental influences that can play a part in the development of GERD. For the purposes of my research paper, I will cover research on many environmental factors, including diet, physical activity, lifestyle choices, and mental health disorders. In this presentation, we will specifically just focus on diet and its connection with the presentation of GERD symptoms. It is important for dietary patterns such as nutrient composition, eating schedules, and behavior to be assessed to understand their role in the development of GERD. A 2019 review by Newberry et al. Analyzed a list of dietary targets, which included food items and eating behaviors that were linked to GERD. Through this analysis, the researchers found that acidic foods and beverages were linked to increased GERD symptoms because they caused direct esophageal mucosal irritation. Coffee, alcohol, and spicier fried foods induced relaxation of the lower esophageal sphincter, which led to vomiting and gastroesophageal reflux. They also found that the timing and size of meals influenced the presentation of GERD symptoms in patients. Late-night meals should be avoided since they cause increased gastric acid production, while large and calorically dense meals lead to increased gastric distension, which is the enlargement and swelling of the belly area. After discussing the genetic and environmental factors that are involved in GERD development, we can now use that information to discuss future research and upcoming treatments that will improve the quality of life and outlook for GERD patients. Let's specifically talk about gene-targeted treatments for GERD, as we spent quite a bit of time in this presentation discussing genetic influences. Targeted therapies are actually cancer treatments that are designed to target the genetic mutations found in a tumor of an individual patient. These targeted therapies are then able to precisely identify and attack the cancer cells, as shown in this diagram right here. The same concept of targeting specific disease-associated genes can also be applied to the treatment of GERD. After identifying a possible risk locus for GERD, researchers can explore prospective treatments that specifically target the identified gene and the protein it expresses, allowing them to target whole genetic pathways through novel drugs. Earlier in this presentation, we discussed how a correlation between the ADAMTS17 protein coding gene and GERD has been discovered. Let's explore an example of a specific targeting mechanism for another ADAMTS enzyme coding gene that can potentially also be applied to ADAMTS17 to inhibit GERD symptoms. A recent 2024 research paper by Maibaum et al. Details the scientists' discovery of the first orally bioavailable ADAMTS7 inhibitor, a molecule called BAY9835, whose chemical structure is visible right here on the right side of this slide. The researchers eventually synthesized a molecule, which was BAY9835, that selectively blocks the function of ADAMTS7. Relevant to GERD, it could be possible that a similar type of inhibitor can be developed for ADAMTS17, with chemical modifications to account for the structural differences. The basic ADAMTS family structure is overall conserved throughout, with the majority of ADAMTS enzymes being composed of the same domains. This similarity in structure will prove to be helpful when developing a similar small molecule inhibitor for ADAMTS17, which could be revolutionary in preventing or reducing GERD expression. Of course, more varied treatments of GERD are also possible. Treatments targeting environmental influences can be incredibly useful and also easy to implement. Lifestyle changes can help prevent damage to the esophagus and reduce the intensity of symptoms present in patients. In fact, diet and lifestyle changes are usually the most common first step suggested by physicians when exploring treatment options for patients. With GERD being a gastrointestinal disease, diet changes are the most crucial steps to take. Dietary changes commonly suggested by doctors include the elimination of trigger foods, which are foods that set off reflux. While these differ from patient to patient, common trigger foods to avoid include chocolate, coffee, and fried or spicy foods. Physicians also recommend that patients avoid larger meals and instead spread out their food intake throughout the day. Physical activity is also beneficial to include as it keeps you at a healthy weight and reduces your risk of any other major illnesses. With regard to genetic influences, enzymatic regulation is also a specific treatment that can be explored. Identifying molecules that regulate expressed enzyme functionality through various mechanisms such as the exposure of catalytic sites can help in the development of specific drugs or biomarkers that provide information on disease diagnosis. It is clear that the investigation into the causes of GERD is quite broad, and further research is necessary to understand the specific causes of GERD development. Further investigation can also yield the discovery of other possible genetic risk loci or environmental influences. For example, a deeper look into the function of ADAMTS17 led me down a rabbit hole to a discovery about the functions of other ADAMTS protein coding genes, such as ADAM10, which is also an established candidate for gene for GERD. ADAM10 controls E-cadherin proteolytic cleavage, which is a biological mechanism that occurs in a higher rate than normal in GERD patients. E-cadherin cleavage leads to increased permeability due to a loss of cell-to-cell adhesion, which is a pathway that has been implicated in GERD pathology. More research on ADAM10 is definitely needed, but further research overall should also be conducted to arrive at more potential causes of GERD. To conclude, in today's presentation, we went over what GERD is, the prevalence and importance of GERD in today's society, and the genetic factors that have been linked to GERD, specifically the genes BARX1 and ADAMTS17. We then also talked about environmental influences that play a part in GERD development, especially diet, and wrapped up our presentation by discussing what's next in the GERD treatment landscape, gene-targeted therapies, other potential treatments such as lifestyle changes, and further research avenues to be explored. I would like to acknowledge my mentor Maria for her invaluable support and guidance throughout this process, and also my family for always supporting me. I hope you all learned something new today and take away an interest in GERD. It's a very important topic, but also really interesting, and it's key to keep having conversations like these so that we can increase exposure and knowledge of GERD and continue to make critical advances in the field. Thank you so much for your time today, and thank you for listening.